Okay, we are going to look at now the chemistry of behavior, neurotransmitters, and neuropharmacology. So let's look at the life cycle of neurotransmitters in this graphic here. And we'll go down the list. Number one, okay, neurotransmitter molecules are synthesized from precursors under the influence of enzymes. So that's how they're built. Number two, neurotransmitter molecules are stored in vesicles. And you are aware of that. We went through the anatomy of a neuron. And so we talked about vesicles. Neurotransmitter molecules may leak from their vesicles and therefore then be destroyed by enzymes. And number four, action potentials cause the vesicles to fuse with the presynaptic membrane with the help of calcium, right? And release their neurotransmitter molecules into the synapse, which is that open space between neurons. And number five, released neurotransmitter molecules bind with autoreceptors and inhibit subsequent neurotransmitter release. The idea here is that there are presynaptic receptor sites, these autoreceptors. If there's too much neurotransmitter out in the synapse, they will find their way back onto an autoreceptor. That tells the presynaptic neuron there is too much neurotransmitter out there. Don't create or release more. Okay, so it's a modulatory process. Number six, released neurotransmitter molecules bind to the postsynaptic receptors. And we talked about what that does. If it opens up an ion channel, sodium flows in. If enough sodium comes in, we reach the point of, the action potential being fired. And that's how the neurons communicate. Number seven, released neurotransmitter molecules are deactivated by either reuptake or enzymatic degradation, and they're torn apart by enzymes. Okay, let's talk about all of the different types of neurotransmitters or their classes now. Okay, so the first one is amino acids, okay, usually found at fast-acting directed synapses in the CNS. What we mean by directed is released by an axon terminal, right? And then picked up by either a dendrite or another axon or a soma. And they are small molecules. So for example, glutamate. Glutamate is the most prevalent of all neurotransmitters and most important excitatory neurotransmitter in the CNS. It is implicated in learning and memory. Okay, aspartate is also an excitatory neurotransmitter. GABA is the most prevalent inhibitory neurotransmitter in the CNS. It's actually prevents seizure activity, and benzodiazepines, for example, Valium, mimic GABA to reduce anxiety and induce sleep. And we also see glycine with an inhibitory role. Okay? The reason why we bring these up, there are many, many other neurotransmitters that we do not discuss. But these are going to be the primary neurotransmitters and the neurotransmitters that you will see in subsequent lectures as we go along, either like in the case here with seizure activities, with Alzheimer's, with various drug interactions, okay? Then we get to the monoamines. Well, mono means one, so we're looking at neurotransmitters that are developed from one amine. The effects tend to be diffuse. They're also small molecules, okay, just like the previous ones. And some examples here, and these are going to be the most common for you to know. So these are really, really important neurotransmitters to memorize. Okay, the catecholamines are synthesized from the non-essential amino acid tyrosine. Tyrosine is internal in the body. Okay? Dopamine, the first one here, involved in motor function, reward, and higher cognition. We will talk a lot about dopamine. Norepinephrine, important for fight or flight response, also alertness and arousal functions. And then epinephrine, okay, important for fight or flight response once again, and increases cardiac output and raises glucose levels. So those are your catecholamines, okay? Dopamine, norepinephrine, epinephrine. Now, also under these monoamines, we have the indolamines, which are synthesized from the essential amino acid, which means you have to get from your diet, tryptophan. Okay. Serotonin, involved in mood, sleep, and anxiety. We will talk about each one of these many, many times. So what I've also provided you here is the abbreviation for each one of these. So you'll notice that dopamine is DA, norepinephrine is NE, epinephrine is also known as adrenaline. So you might see that in the book. And serotonin, okay, is SE. So as you read journal articles, textbooks, and so on, sometimes they'll only provide you with the abbreviation SE or BA. So it's good to memorize that. What I also provided you were the names of the receptor sites, okay? So the receptor for dopamine is called dopaminergic. Okay. The receptor for norepinephrine is noradrenergic. The receptor for epinephrine is adrenergic. And then finally, for serotonin, those are called serotonergic. Okay. Also, we have acetylcholine, which is also a small molecule, just like everything that we've discussed so far, involved in the restful response of the peripheral nervous system, PNS, important for muscle contraction, involved in memory, and widespread loss is implicated in Alzheimer's disease. And so I did the same thing here too. The abbreviation I provide you, ACH for acetylcholine, and then those receptors are called cholinergic receptors. We also have some unconventional neurotransmitters. Again, they're small, okay? But here the examples would be soluble gases and then your endocannabinoids, okay? The soluble gases, these have no receptors. They diffuse through the membranes. I give you the example here. They can even pass from postsynaptic to presynaptic. They don't follow the same rules that we've seen with these other neurotransmitters. One example would be nitric oxide. It increases blood flow and lowers blood pressure. And carbon monoxide, which is involved in vasodilation and circadian rhythms. And there's a lot of study, much less are known about the soluble gases than we know, for instance, the catecholamines, okay? And then the endocannabinoids are endogenous internal cannabinoids, okay? One example here is anandamide, which is not well understood, but seems to be involved in pain relief and inflammation. And we will definitely go over these in further subsequent lectures. Okay. And then there's the neuropeptides. Now, for the first time, we're seeing these as not small molecules, but large molecules, okay, made from many amino acids. Examples here, endorphins, the endogenous opioids, our internal opioids, produce analgesia, pain suppression. We see oxytocin involved in childbirth and breastfeeding, and substance P involved in pain perception, memory, and cell growth. And there are many others in this. I'm only providing you a small sampling. This graphic now shows you pretty much everything that we went over, but all in one place for you. So you'll notice how the amino acids, the monoamines, acetylcholine, unconventional neurotransmitters, all small molecule neurotransmitters, and then your neuropeptides are going to be the large molecule neurotransmitters at the bottom. Okay. I also wanted to show you the pathways for these systems, okay? So on the bottom left, again, now we get to use our terminology here. So we see cholinergic. So cholinergic means that it's going to be dealing with acetylcholine. And you'll notice that the pathways run from the basal forebrain to all of those other areas. Okay, so, and there's lots of prefrontal cortexes on there. We see it going up to the motor cortex, frontal lobe. Okay, and then next we have the dopaminergic. So here we're dealing with dopamine, very important in the mesolimbic cortical pathway. the ventral tegmental area, and then also what we call the mesostriatal pathway. Okay? So implicated in movement, implicated in reward. Okay? Noradrenergic, okay? Norepinephrine. Here we're looking at areas like the lateral tegmental area, okay? And moving on to the brainstem and spinal cord, or the locus ceruleus to the forebrain. And then lastly, you have serotonergic. So this is going to be a pathway for serotonin from the midway structure raphe nuclei to the forebrain. Okay, now, since we know a little bit more about neurotransmitters now, let's talk about neuropharmacology. The idea here is that psychopharmacologists, okay, are going to be scientists that are developing drugs that can pass the blood-brain barrier, enter into the brain, and then interact with these neurons, okay, either to create an agonistic effect to increase an activity, cause more of something, or an antagonistic effect, which decreases or inhibits activity. Okay, that's what they're looking at. And the more selective we can create these medications, these molecules, the better off we are. Because the issue is that, and you've probably seen these on commercials and so on, you have a drug that works for one thing, but has all kinds of side effects, or what they call contraindications. Okay, and that's because primarily we're not looking at these drugs being very selective. Okay? When they enter in, they pass the blood-brain barrier, they can affect the brain in any area. Okay, now we do have some that are selective to specific neurotransmitters, which is fantastic. We have some that are specific to certain receptor sites, which is also fantastic, but they're still affecting the brain kind of overall, all areas of the brain. It's not centralized to one area of the brain. Okay. So let's look at both of these mechanisms. On the left-hand side, you have agonistic drug effects. Okay. So in this case, a drug might be created to increase the synthesis of a neurotransmitter molecule, making more of it. So let's just say that we stick with dopamine. Okay, there might be a drug created to increase the synthesis of dopamine so that we have more dopamine. There may be a drug that increases the number of neurotransmitter molecules by destroying degrading enzymes. So the drug is actually an enzyme, or the drug, sorry, is something that destroys the enzymes so that the enzymes cannot break dopamine down. Okay, so it's still creating more dopamine and being an agonist. The drug increases the release of neurotransmitter molecules from the terminal buttons so that there's more dopamine out in the system. The drug binds to autoreceptors and blocks their inhibitory effect on neurotransmitter release. So there might be a drug that's developed, okay, to bind to that autoreceptor. Remember what that does. Now, the presynaptic neuron is going to continue to release more neurotransmitter because it's not this feedback, it has been blocked. The drug binds to postsynaptic receptors and either activates them or increases the effect on them in the neurotransmitter molecules. So this is where you might hear that the drug mimics. Okay? So a drug is created to mimic dopamine. So it fits into the dopaminergic receptor site and causes that ion channel to open, etc. Or the drug blocks the deactivation of neurotransmitter molecules by blocking degradation or reuptake. So the drug is created so that it blocks the reuptake channel there, or it destroys the enzyme in the synapse. Okay? Both of those things causing more of the neurotransmitter or more of the dopamine in the example that we're doing here. Now, if we look on the right-hand side, okay, maybe it's not the best situation for us to create a drug that is an agonist, but maybe we have to create a drug that is an antagonist, okay? A drug that blocks the synthesis of neurotransmitter. So if you're blocking the synthesis of the neurotransmitter, you're going to end up with less of it in the system. The drug causes the neurotransmitter molecules to leak. And if they leak from the vesicles, then they're destroyed by enzymes. The drug blocks the release of the neurotransmitter molecules from terminal buttons so they can't leave the presynaptic neuron. The drug activates autoreceptors and inhibits neurotransmitter release. And then finally, the drug is a receptor blocker. So it binds to the postsynaptic receptors and blocks the effect of the neurotransmitter, which means that it attaches to the receptor site and it causes that ion channel to maybe not open. And if it does not open, then you can't get that sodium to flow through, and therefore the postsynaptic neuron does not become less negative, and you have the chance for the action potential. Okay, so this is the field of neuropharmacology, is to have a very, very solid understanding of what is going on at the neuronal level, and then trying to identify what could be done through these different medications to alter this system, to either increase the amount of neurotransmitter or decrease the amount of neurotransmitter, depending on the situation. And then I just wanted to leave you with some examples here. Neuropharmacology is the study of exogenous and endogenous chemicals on neurobiology. And some examples now, you might be familiar with the SSRIs. These are selective serotonin reuptake inhibitors. So these are drugs that increase the amount of serotonin in the system. And think about the terminology here, guys, okay? So selective means they only act on serotonin. That is a major, major accomplishment for neuropharmacology, being selective, okay? So it only affects serotonin, and they are reuptake inhibitors. So if the serotonin is not being reuptaken into the presynaptic neuron, then it's staying out into the synapse. And guess what? Then it's more likely to fit onto a receptor site of the postsynaptic neuron and then cause that communication to go to continue down the line. So it is an agonistic effect. SNRIs, okay, selective norepinephrine reuptake inhibitors. So these are drugs that increase the amount of norepinephrine in the system, also an agonistic effect. Opiates work to mimic endogenous opioids, providing pain relief. And then, like I said, these are just some examples for you. And also dopamine antagonists are used for the psychotic symptoms of schizophrenia. We could go on forever and ever about other different examples, but I just want to give you an idea of what we're looking at here for neuropharmacology.